was very moved to see that this hall is named after Prof Bhargava, who was a dear friend and a mentor for me for many years at that time. So this is something that this visit especially has been very, very uh, emotional for me because I have been coming here. In fact, Bhargava wanted me to join CCMB, and at that time I had other options, so I did join. So it's delightful. I'm delighted to be here. Uh, I have called this War and Peace not because of the war that's going on right now, but because of the war that might go on in these insect societies. I will begin by giving a 5-10 minute introduction to insect societies before I talk about our own research. I think almost everybody is familiar with honeybees. So honeybees are sort of the prime example of insect societies. You can say they're the quintessential insect societies. As you all know, honeybees live in large colonies uh, around a nest, and a colony consists of maybe 10 to 60,000 individuals, all in this crowded space. And they organize one of the most efficient societies, and it's very, very interesting to understand how they actually do so. This particular picture is of interest because this is a very unusual kind of honeybee. There are some 15 species of honeybees in the world. This is one of the smallest ones, and it's called the Asian dwarf bee. It's our local bee, and amongst all the honeybees, this one is slightly more primitive in its social sophistication, and that makes it very attractive for us to study it because all others have already reached what you might call the pinnacle of sociality. This one is still getting there, and therefore it's very interesting to study. This is called Apis florea. This is a picture taken from IISc. This is a picture of close-up of the queen bee and the workers. So a typical colony of honeybee will consist of one and only queen. She is a large, fertile, the only mated individual in the colony, and all eggs are laid by her. The rest of the colony consists of a small number of males whom we call drones. Now, for interesting reasons, the males do not participate in social life. They do not participate in domestic life. They are proverbially lazy. In fact, we use the word lazy drone, and I won't talk about males in this talk for sure. The rest of the colony consists of smaller females who are nearly sterile. And these are the ones we call workers. And what you see here is a picture of the queen surrounded by a group of workers. Now, the queen does only two things. She lays eggs and she produces chemicals, which we call pheromones, which come out of her body and influence the behavior of the rest of the colony. These are only two things she does. But in order for a colony to function, many things need to be done. The nest has to be built, it has to be repaired from time to time, food has to be processed, larvae have to be fed, the nest has to be cleaned, the nest has to be guarded, food has to be brought from outside. All of this is done by the sterile workers. And they do this in a very orderly fashion. In fact, what a bee will do depends on its age. The youngest bees will be cleaner bees. When they grow a little bit older, some glands in their abdomens will develop, which are called wax glands. And using these wax glands, they convert the nectar that they have drunk from the flowers into wax, secrete the wax outside, and build this beautiful hexagonal cell. So they are builder bees. A little later, other glands in their mouths will develop, which produce other chemicals which are required to feed the larvae. So they become nurse bees, and then they become the food processing bees. Then they become guard bees, and eventually they go out and forage and bring back food. Now, what you see here, just a few snippets about the social life of bees. So you see here this queen who is distinctly larger, and she's surrounded by a small group of workers. If you watch this carefully, you will find that these workers are on a special royal duty. Their job is to take care of the queen. They clean the queen, they lick the queen, they feed her because she has no time to do these things. She's busy laying eggs. She can lay thousands of eggs per day, so she's really busy. But as she moves, the whole retinue moves along with her. If you watch this a little longer, you'll be surprised to find that these workers who are so diligently taking care of the queen, within a few minutes, they seem to lose interest in their job and go off and do something else. Other bees who are doing something else will come and replace them. But if you think about it, there is a great advantage of such short shifts of work, because the workers, when they are in the vicinity of the queen, they absorb the chemicals she has produced, and when they go off elsewhere, they pass these chemicals on to others, and other bees who now come to close to the queen can get the chemicals. So the chemicals that the queen produces get disseminated into the colony very efficiently because of this turnover of those workers who are taking care of the queen. So just one interesting snippet. What I'll show you here is that honeybees, of course, are not the only insect societies. Ants are equally interesting, perhaps in some ways even more impressive. If there are 15 species of honeybees, there are at least 15,000 species of ants, and probably at least 15,000 more waiting to be discovered. Now, I already mentioned that honeybees change their task as they grow old. Now, ants also are social. They also have one large fertile queen, a few lazy drones, we don't call them drones, but they are males, and a large number of sterile or nearly sterile female workers. In the case of ants or many ant species, the work that an ant does is not dependent on its age, but it depends on the shape and size of its body, which means ants are born with a shape and size that is appropriate for the task they should perform. So these ant subgroups are already morphologically differentiated. Here, for example, is a rather famous drawing of different ants present in a single colony. Now look at this individual and compare her with this individual. They look so different, but their body size and shape and structure is suited for the tasks they perform. For example, this ant would be very efficient at carrying small seeds and bringing it back to the nest, a task at which this ant would be hopeless at. Whereas this ant would be a very good soldier, able to attack enemies and guard these row of small ants that are coming to the colony. So this kind of division of labor happens in the honeybees without morphological differentiation, in the case of ants with morphological differentiation. Now in the case of ants, it's rather interesting. Because of this change, there is a great deal of morphological variation between the same colony. In fact, ants hold the world record for intra-species, intrasexual physical dimorphism. And the world record is held by this Asian ant, which occurs in Asia, and this particular thing was studied in Malaysia by a friend of mine, Mark Moffat. And what Mark Moffat has done is he's taken the largest member of the colony and placed on its head the smallest member of the colony and taken a scanning electron micrograph. It turns out that the largest member of the colony weighs 500 times the smallest member of the colony. And I like to remind my developmental biology friends that both of these come from the same genome. So the differentiation is really entirely epigenetic. This is a picture showing ants in action. This is a very common species of ant. You will see it all over here. This is what we call the weaver ant. There are two species. There is an African species and there is an Asian species. This particular photo is of the African species, but we have a very similar species here. And these ants, we call them weaver ants because they build nests with leaves on the trees. Now you can see here that there is a leaf here and there is a leaf here, and a large number of ants are working very hard to bend these two leaves and align them. So that if they align them, they can actually stitch them together. And what do they stitch them with? Nothing less than silk fibers. They use silk to stitch these leaves. Where do they get silk from? Adult insects do not make silk. Larvae make silk. So when these ants succeed, as you can see, this is a very interesting coordinated effort. They make these large number of bridges between these two, and slowly the ants in the middle will drop off until these two leaves are aligned in this fashion. And then what will happen is one of the ants will go inside the nest, pick up a fully grown larva, bring her outside and squeeze her appropriately and persuade her to donate some silk for the community good. And you can see the silk fibers here through which they actually stitch these leaves. I just want you to imagine the amount of cooperation, coordination that is involved for the ants to pull off such a feat. You're all familiar with termites. So termites are another example of a highly evolved insect society. This is, of course, the mound that you see outside. Most of the action happens underground, and here is an artist's conception of what a termite colony might look underground. For each group, I want to tell you something unique that they have done, at least one unique thing. For the ants, I told you they are morphologically differentiated. The unique thing about termites is that unlike ants, bees and wasps, by the way, the ants, bees and wasps, as I told you, the males do not participate in social life, and therefore they are called feminine monarchies. Whereas termites are the only ones in which the males have an equal role. You have a queen and you have a king. You have female workers and you have male workers. In fact, in this colony, you will find that this is the queen. Now, her abdomen is swollen because it is full of eggs. When she began her life, it wasn't swollen, but as she produces more and more eggs, she gets swollen, and this is the king. And what you see next to her, this is the king. He's not very impressive, but he is indeed the king. The king and queen live together in this royal chamber, and they continuously produce offspring. So that is something unique about termites. That brings me to the fourth and more or less the last group of highly evolved insect societies, and these are the wasps. I'm particularly fond of wasps, as you know, because I work on wasps all the time. There are many thousands of species of wasps in the world. Not all of them are social. Many of them are solitary or parasitic, but a few thousand species are social, and nearly all social species of wasps can be called paper wasps. And the reason why they can be called paper wasps is they build their nest not from wax as honeybees do, not from leaves as ants do, not from soil as termites do, but from paper. Now, where do they get manufactured? Where do they get paper? They manufacture paper. How do they manufacture paper? Exactly as we manufacture paper. They scrape cellulose fibers from plants, chew it up, add various chemicals, make it into a fine pulp, spread it into a thin layer, and dry it by fanning their wings. Indeed, it is paper, you can write on it. This is all paper. If you take it out, you can write on it. The entire nest is made from such paper which they manufacture. And this paper has very interesting properties. It is resistant to rain. It is very hardy. They make this paper. Now, don't try this at home, but I once opened this envelope and I found what I like to call a multi-storied apartment complex. So the wasps have built many layers of comb in which they rear their brood. And if you turn this around, this is what you see. These are all the pupae of the wasp. And like in the honeybee case, you can see these perfectly hexagonal cells. And a major difference between honeybees and wasps is that honeybees use these cells to rear their young ones as well as to store food. In the case of wasps, only to rear their young ones, not to store food, because wasps are non-vegetarian. They are carnivorous and they have not invented a refrigerator. So they cannot store food. They have to find their food every day. But these cells are used for rearing the young ones. Many people say, yeah, this is wonderful, but why do you really want to study them? Is it worth studying? And today, of course, you know that we all have to tell us what is the use of the work that you are doing. People refuse to read a wonderful little book that I read every now and then. It's called The Uses of Useless Knowledge. Now people are not interested, so everybody is interested. So I want to assure you that there are great advantages of studying insect societies. The answer to the question, why do you study insect societies? There are myriad applications. Applications in pharmacology, physiology and medicine, telecommunications and division of labor, decentralized work organization, even robotics and artificial intelligence. All of these fields have progressed rapidly because of studying insect societies. But of course, it's not just that. There is also sheer curiosity. At least for me, I say, how can we possibly turn away from little insects fighting for a territory, sacrificing themselves to save each other's lives, practicing agriculture, caring for the injured, building structures with exquisite architecture? There's an endless list of fascinating things that they do. There is a third aspect, which is more philosophical, and that is animal societies can hold a mirror to us to understand ourselves. Now, so there are at least three broad kinds of reasons why we should study insect societies. But for me, curiosity is adequate. And if you think that makes me less patriotic than somebody who is doing applied research, let me assure you that every one of these applications had their origin in curiosity-driven research. I can actually pinpoint Every application that has come from the study of insect societies to all of these fields began because somebody was curious. Why does this ant do that? How does this bee get here? So curiosity-based research is the mother of all applied research. So we must pursue both simultaneously or sequentially. But if you want to do it sequentially, curiosity-based research has to come first. You can't have the applied research coming first. If when the field matures a little bit, you can do it parallelly. But if the species, you have to begin with curiosity-based basic research. Only then applications will follow. So I, of course, work purely out of curiosity. Now, I showed you a large wasp which covers her nest with paper envelope. I do not study that wasp. In fact, that is the only wasp I dislike. And that is because that wasp is a predator of my wasp, the one I study. The one I study is much more. The power is not connected, is it? It's okay? Can I just continue? No? No? It says 3 minutes to restart. It work now? Okay. So this is the species I study. This is what I like to call the Indian paper wasp. Its scientific name is Rhopalidia marginata. The nice thing about this is, first of all, the colonies are small. They are not very aggressive to us, easy to study them. Most important, they do not cover the nest with envelope so that I can watch, I can see everything. The nest is more or less a two-dimensional structure. So everything is at one plane. You can watch. You can see there are adults here, there are pupae, there are larvae, there are eggs. All of this can be studied. All interactions can be studied. You can study it manually. You can film it. You can study it in many different ways. And so this is a wonderful species study. I spent most of my life studying these species. In fact, I first came across this species when I was a first year undergraduate, and since then I've been fascinated with this species, and I continue to work with this species. There are only two major things that I had to do to make it possible to study this. The first is that I needed a way to identify, distinguish one wasp from another. As you can see, they all look alike. And then you can't make much sense. But it turns out that it is very easy to do that. We in fact have perfected methods of marking every wasp with unique spots of colored paint on different parts of the body. And every wasp then has a 5-letter name, and that is in name is in the computer. So every wasp has a file with a 5-letter name, and we keep on adding information about each wasp in our computer files. So we have dossiers for hundreds or maybe even thousands of wasps in our computer. So this is one major advance that is required. The second is how to study them or where to study them. It turns out that luckily this species likes to live around human habitation. So we don't have to go very far. Often the wasps come and build their nest in my lab or in my house. I don't have to go very far. So it turns out that you can study them wherever they are. But it is also very easy to transplant them to the laboratory and put them in cages. So we study them both where they occur as well as in laboratories. And we have a very nice laboratory, except that this laboratory has no walls. All that this laboratory has is wire mesh all around. And the purpose of this wire mesh is to allow my wasp to go in and out, but prevent that big wasp, the Vespa tropica, the one I showed you, which eats this wasp, prevent it from coming inside. And Rhopalidia wasps can be reared in little plastic boxes like this, which I steal from the kitchen, or smaller box size, or indeed very large boxes in which I can put a wasp nest and a student inside. All these are possible for study. So that's really all you need to study these wasps. And what did I want to do? When I set out to study these wasps in the beginning, I said I want to understand everything humanly possible about this species. That was my goal. And what I'm going to tell you today is some extracts of what we have been doing. So this is Rhopalidia marginata. There are many themes that can be pulled out of the research. See, I've been working on this, as I said, ever since I saw them as a first year undergraduate student. So I want to pick up one theme, and that is the theme of cooperation and conflict, or war and peace, as I've called it today. But before I get into the details, I want to digress for a moment and tell you some of my pet ideas. I find that the problem with scientists is, especially when they communicate, is they do not talk about the process. I believe that the process of science is more important than the product. And the reason is that the process by which you have found something may be of interest to a large number of people and the product only a few people. More importantly, if you communicate the product and do not tell the process how you got the product, science sounds like magic. This creates a barrier between science and scientists and the public, scientists and funders, scientists and politicians. We seem to hide the process. It's very important for us to convey the process. Sometimes we feel that the public cannot understand, and that's not true, because if they cannot understand, the fault is ours, not theirs. So I find it very important to communicate the process of my science. So in this talk, I will tell you as much of the process as of the product, maybe a little more about the product process than about the product. So let me tell you how we do our research. First and foremost, one must love the species one studies. And we love this species. I and all my students are fascinated. We can sit for hours and hours and hours and watch these wasps just for fun, especially if the wasps are marked. It is impossible. I challenge anybody to get bored in front of a wasp colony with marked wasps, because it's so fascinating. You're always, something exciting is happening there. So we basically watch them for fun. That is our real goal. That is the primary goal. We want to have fun watching these wasps. But of course, when you watch these wasps, what happens is that since we are thinking people, questions arise in your mind. It doesn't have anything to do with our being scientists. It doesn't have anything our having been trained. As human beings, especially since we are social beings, questions arise in your mind. How did this wasp manage to do such and such a thing? Why did this wasp do such and such a thing? Many such questions will inevitably arise in your mind when you sit and watch them. And then you will realize that most of these questions that arise in your mind are probably not easy to answer. But every now and then a question arises in your mind, you think, I might be able to answer that question if I did such and such an experiment, if I did such and such a piece of research. And when that occurs to us, then we sit down and say, let's now have a research program. We design an experiment, we plan everything. Now we become scientists and we plan. And we actually perform those experiments. Those experiments may involve manipulation, may involve more observation. And then we seem to have an answer. For a moment, we are thrilled. But immediately we are crestfallen because we think this answer cannot be correct. It probably is not right, because the answer raises in your mind many new questions. And unless those questions are answered, you have very low confidence in your answer. Now, there are two very happy consequences of this. First is it motivates you to go to the next, answer the next question immediately, nobody has to tell you. The second is it injects in your mind caution. And caution is very important doing this. So then we go and try to answer other questions. When you answer the next question successfully, your confidence in the answer to the first question goes up one notch, but for the second question answer, you still don't have confidence. For that, you have to answer the third question. So our research is a series of questions and answers, and this talk will also be a series of questions and answers. I've just submitted a book manuscript illustrating some 35 such questions and answers. I will take some 7 or 8 and illustrate them in this talk. So this talk will actually be a series of questions and answers. But I should not forget the bigger picture. We said war and peace. So I would like to begin with some stories of peace. That's just my nature. I'd like to begin with peace, and we'll come to war in the end. When I watched these for the first time, the first question that occurred to me is, unlike in a honeybee colony, unlike an ant colony, the queen is like everybody else. In this species, there is no morphological difference, not only between workers doing different types of work, or even between the queen and worker. In fact, in this colony, this is a colony of wasps. One of these is the queen, and there is no way you can tell which one it is. Even I cannot tell which one she is. I have to watch patiently, wait for her to lay an egg. When she is laying an egg, then I know that she is the queen. Otherwise, I have no way. So she is like everybody else. And yet, in every colony, there's only one queen. Only one individual functions as a queen. Only she lays eggs. Nobody lays eggs. Everyone respects her. Everybody works. How is this possible? She doesn't look any different. So obviously, my very first question was, how does the queen behave as compared to her? She doesn't look different, but she must be behaving differently in some way. Otherwise, why would the workers respect her, recognize her, or even understand who she is? So I wanted to understand the behavior of the queens and workers. And luckily, I was not trained in this field. I was not trained as an entomologist. As Janvi mentioned, I was trained as a molecular biologist. So I used common sense. And this has been very helpful for me. In fact, I believe that science should always begin with common sense. And only when common sense fails, one should try to do something else. Otherwise, common sense is the best starting point. So using common sense, I took out a notebook and a pencil and I started writing down in plain English, what do the wasps do anyway? What is what they do? So I got entries such as a wasp is sitting, a wasp is walking, a wasp is flying, a wasp is feeding the larva, a wasp is receiving food from somebody else, a wasp is biting another wasp, a wasp is chasing another wasp, a wasp is repairing the nest, is building the nest. So I started making in plain English, in common English words, a list of behaviors. And as I watched and watched, the list grew. And I had about 100 such items in my notebook. Now I said, aha, now I have this 100. I want to know which one the queen does and which ones the workers do. Then I'll know how the queen behaves differently from the workers. So I now watched more carefully with reference to each of these entries in many colonies. And I found to my great surprise that the queen can do all the 100 and the workers can do 99. The only thing they cannot do is to lay eggs. So there is no difference between the ability of the queens and the workers. So I hypothesized that if they can all do everything, maybe they don't do everything to the same extent. Maybe there is a quantitative difference between queens and workers in their behavior rather than a qualitative difference. So this was early 80s. I developed methods to measure their behavior. And I measured their behavior in two ways. One is I calculated the proportion of waking hours which each wasp spends in each behavior. How much time does a wasp spend sitting? How much time feeding? How much time fighting? How much time going out? So this we call it a time activity budget. So I budgeted their time and made a budget for that time. Now, some behaviors are very, very short duration, like biting somebody, pecking somebody. Those, it's not easy to measure the time because it's in seconds. So those I measured in a different way. How many times per hour do you do this? How many times does wasp A bite wasp B? So I had measured behavior in two ways: proportion of time spent in that behavior, number of times that behavior is performed per unit time. So I made these measurements for all the 100 behaviors for lots of wasps. Now, as you can imagine, I had a huge data matrix, large number of wasps, large number of behaviors, and in the cells I had information about either about the proportion of time spent in that wasp behavior by that wasp or the number of times that behavior was performed by that wasp. Now, this kind of data is obviously ideal for statistical analysis, in particular for multivariate statistical analysis, because I wanted to look at all the behaviors simultaneously. So I performed two kinds of multivariate statistical analysis. One of them is called principal components analysis, which allows you to reduce the dimensionality of the data and make it easier for you to visualize and handle the data. The other is called a cluster analysis, which tells you whether there is any pattern in this. And applying these two things, again, I'm going to make a long story short, we found something rather surprising. We found that the wasps in each colony can be classified into three types based on the quantitative measure of the behavior. So you can see here there are three rather distinct clusters. After I got these clusters through the principal components analysis and cluster analysis, I went and looked at these clusters in greater detail and asked, what do these wasps do? It turns out that one group of wasps spent a very large proportion of their time just sitting and doing nothing. So obviously I called them sitters. There were another group of wasps which were much more active in aggression, in fighting, in biting, in chasing other wasps. So I called them fighters. A third group were really the honest workers. They would go out of the nest, bring food. They did a lot of hard work and they went out foraging. So I called them the foragers. So we showed that in each colony, wasps can divide into three groups, which we call sitters, fighters, and foragers. In insect societies, when subgroups of members of a colony do something, we call them a caste. So you have queen caste, worker caste, forager caste, nurse caste, soldier caste. We use this word caste for social insects historically. Now, historically, these words are only used when there's a morphological difference. Now, I have no morphological difference, so I coined the phrase behavioral caste. So these are three behavioral castes seen in this species. Mostly when people study insect societies, the queen in all other species looks very different. She's big, she's royal, so they give her special attention. They treat the queen differently, they study the worker differently. Before you study, you already know that I'm studying the queen or I'm studying the worker. In my case, I said, since the queen looks like everybody else, why should I give her any special treatment? So in most of my work, I didn't even care who the queen was and who the worker was. In other words, I did not tell my computer who the queen was and who the worker was. And that was a great lucky thing, because now after getting this classification into three groups, I could go back and post fact to ask, where is the queen? Is she a sitter? Is she a fighter? Is she a forager? This question I could ask because I had not worried about it beforehand. And back to common sense. Here's an example where you have to go beyond common sense. Common sense said the queen must be a fighter. Because in other such species which had been studied previously, it was shown that in small colonies like this, where queens are not morphologically different, they rule by physical aggression. Literally what they do is they constantly physically harass their workers and make them work. If a worker is sitting doing nothing, go and poke her, go and prod her, then she'll go bring some food, deliver it, and again go back to sleep, then you go and prod her again. In fact, it had been shown that if you remove the queen from a colony, all the workers will just become lazy and do nothing. And the queen has to push them. Also, the queen uses physical aggression, literally physical harassment, to prevent the workers from becoming future queens. So both preventing them from becoming future queens and making them do real work is the responsibility of the queen, who does this through actual physical aggression and activity. So obviously our queen must at least be one of the fighters. There are more than one, but she must be a fighter. And since we did not do this beforehand, now when we ask the question, lo and behold, the queen is never a fighter in our species. In colony after colony, our queens are lazy sitters. There is nothing better than finding a result which defies all previous knowledge. That's the best thing that can happen to you. So don't say my experiment failed. My experiment succeeded, because my wasps have not read the textbooks, they have not read the papers, they're completely different. So it is remarkable that the queen in this species, the queen in Rhopalidia marginata, is a remarkably non-aggressive, non-interactive, meek and docile sitter. Now your happiness lasts for a few seconds. Yes, I have an answer, but then how is this possible? It cannot be true, because immediately questions arise in your mind and you begin to wonder whether you have done something wrong or you missed something. For example, if the queen is such a meek and docile sitter, how did she become a queen in the first place? Why do the workers respect her? If there's a strong bully fighting and chasing everybody else, it's easy to see why the workers will say, okay, you are my queen. But if this is a non-aggressive, non-interactive queen sitting somewhere in the corner, why should they respect her? Why should they not develop their ovaries and become future queens? Why should they even work in the first place? So you can see there are great hurdles to accepting the answer. And therefore, you put it on hold and you say, now I'm going to try and answer this question with the hope that I will get some confidence in my first answer. Now, how do we answer this question? Simply staring at them some more will not help, because we have stared at them for a long time. We have observed for a long time. We even measured their behavior. So now you have to do an experiment. Now your common sense says your common sense is wrong. So something special is happening here. So we had to do an experiment. So then we found out that in the life of a colony, see, when a honeybee queen dies, a worker cannot become a queen, because there she is born as a worker. In this species, because they're all identical, anybody can become a queen. In fact, we found that if the queen dies, one of the workers will become a queen. So we said, that's a good thing. Now what we need to do, we need to study a colony when in the process of somebody becoming a queen. because we want to know how does she become a queen in the first place. So that's actually easy to do. All you have to do is experiment, first study a colony, experimentally remove the queen and wait for one of the workers to become the queen and see how this process happens. That will help us to answer this question. So we designed an experiment which lasted 3 days. On day 1, we had a normal colony with the queens and the workers all individually marked and all studied for the whole day. And studied means what? You have this data matrix with proportion of time spent by each wasp in each behavior, number of times each behavior is performed by each wasp. You get this data over a whole day period. Early next morning, before the wasps sort of wake up, you take a forceps and quietly remove the queen. And then the wasps wake up and realize they don't have queen, you study them again for the whole day, which means again you get another data matrix of the time activity budgets and the frequency of each behavior in the absence of the queen. That will tell us how this happens. Now, when we did this experiment first time and when we removed the queen, we were not quite sure what to do with this queen. She doesn't look very different, but still we said we should give some respect to her. We put her in a glass vial, we gave her a drop of honey and kept her quietly. At the end of the day, she was fine. So my student came and said, what shall I do with her? And I said, well, the experiment is over. Now, why deprive her of her colony? Put her back in the colony. But then we said, what will happen if we put her back in the colony? I said, that's interesting. So why don't we extend the experiment to a third day? So introduce the queen and now study the whole boss again with the queen, and the queen has come back. So we had a 3-day experiment with queen, without queen, and the queen returned. And when we did this, the most spectacular result happened. This species is a fairly peaceful species. Yes, there are fighters, there's some biting, some chasing and going on, but on the whole, it's a very peaceful colony. But as soon as we removed the queen, that is, the wasp woke up and realized they didn't have queen, all hell broke loose. The reasonably peaceful society became a hyper-aggressive society. Now, the aggression is in the form of behaviors such as biting, chasing. Sometimes a wasp will hold another wasp antenna and immobilize her for minutes together. And when she releases that, other wasp will run away. Or they'll go and peck at these wasps. Or they'll go and sit on the wasp. So there are about 10 different things they do to each other. 10 different relatively nasty things they do to each other. And this is the kind of aggression they show. But none of this causes injury. It's more symbolic. But in each of these, it's easy to see who is the aggressor and who is the victim. The one who bites is the aggressor, the one who gets bitten is the victim. So we use the word dominance behavior and subordinate behavior. The one who bites is the dominant wasp, the one who gets bitten is the subordinate wasp. The one who chases is the dominant wasp, the one who gets chased is the subordinate wasp. So we list, because we have calculated how many times per hour every behavior is done. We take all these 10 aggressive behaviors and we add them up and we have a score for dominance behavior. How many times per hour does wasp A show dominance behavior overall, or how many times does wasp A show towards wasp B, and so on. So we can look at this dominance behavior. Now it turns out that the rate at which dominance behavior was shown in the colony in the presence of the queen, that rate doubled, quadrupled, 10-fold, 20-fold, 30-fold increase in some colonies immediately after we remove the queen. So a peaceful colony became a hyper-aggressive colony. This itself was a great surprise for us. Even greater surprise was that all of this new aggression, which was not shown on day 1, was by one worker only. One and only worker became hyper-aggressive in the absence of the queen. What happened when we returned the queen? I'm glad we extended the experiment one more day, because something very unexpected happened. You return the queen, this hyper-aggressive worker stopped being aggressive and went back to work. This is what the data would look like. So this is dominance behavior. This is day 1, very little aggression. Day 2, you can see huge amount of aggression by one wasp. And day 3, that aggression is almost gone down. I want you to realize that this is the plot for the queen, this is for this worker who became very aggressive, and this is the average worker of the remaining. I want you to appreciate that these data points we could only calculate post facto. Because on day one, we didn't know who the aggressive individual will be. After we found out that this individual is hyper aggressive, we went back to the computer and asked, what was she doing yesterday? That is this graph. And this is what happens here when the queen is returned. Again, this is an average worker. Now the question is, what happens if you do not return the queen? If you do not return the queen, this hyper aggressive individual will rapidly lose her aggression. So you can say day 2, 4, 6, and by the 6th or 7th day, her aggression is almost zero. And what is most surprising is, at this point, she is actually the new queen. She actually lays her first egg. Because now she is a meek and docile, non-interactive, non-aggressive queen. So there was this phase for about a week in which she was hyper-aggressive. So and this is done only by one individual. So obviously we called her the potential queen. So the answer to our question, what was our question? How, if the queen is such a meek and docile, non-aggressive, non-interactive, how does she become a queen in the first place? The answer is potential queens begin their careers as very aggressive individuals and only later become meek and docile. So you have some reason to think why the workers would respect her. So this raises your confidence in the previous answer, that the queen is in fact a non-aggressive individual. But this question answer itself is very suspect. Because how can this be true? She is aggressive only one week. What happens afterwards? How does she make sure that the workers don't revolt and become queens? How does she make sure that they stop working? Because after a week, she is no longer producing. So obviously we had a difficult question. If the queen is such a meek and docile citizen, how does she regulate the activities of workers? How does she do this? I don't want to give you the impression that we ask a question and we know what the experiment to do is. We do the experiment and we get an answer and we get another question and we know what experiment to do. This is very idealized and is not in congruence with reality. Very often we are just stuck. We don't know what to do. Now, how do you answer this question? You have no clue. There was no obvious experiment to do. So what do we do when that happens? Go back and watch the wasp for fun. That's the default option that we have. And start thinking about it, or maybe I think. Now it turns out, but of course, you see, now when you're watching the wasp for fun, this question is in your mind, it's bothering you. You are no longer just innocent person watching the wasp. You have some question that is bothering you. What might be the reason? How might this happen? And then we came up with an experiment. We said, let us now pay special attention to the work of the wasps and see how the queen is doing this. So we had to choose. They do lots of work, right? They do many different kinds of things. So for the sake of the research, we decided to choose two examples, two important kinds of work that the workers. One important one is to go out and bring food. The other is to feed the larvae. The larvae are helpless. They have to be fed in their mouth. So these two pieces of work are very important. So let us measure the rate at which these workers do these tasks in the presence of the queen and in the absence of the queen. So again a 3-day experiment. Day 1 with the normal colony, measure the rate at which workers bring food, the rate at which they feed the larvae. Day 2 remove the queen, measure the rate at which workers bring food, feed the larvae. Day 3 return the queen and make the measurements. And we may be able to get some idea of how the queen regulates the activities of the workers, even though she is a non-aggressive individual. And again, to our great surprise, most of the experiments, the answer that we get is completely surprising. It's something that we would not have expected. To our great surprise, we found that the workers don't care for the queen. They don't need a queen. They will bring food and they will feed larvae at the same rate, whether the queen is present or not. Rates of feeding, rates of bringing food, day 1, day 2, day 3, no statistical difference. Feeding larvae, day 1, day 2, day 3, no difference. So the workers don't need the queen at all. They don't care for the queen. So what is our answer to our question? Our answer to our question is Rhopididae marginata queens do not regulate the activities of their workers. It's a fair answer, but it opens up a new question. Then who does? Somebody should know, do. Somebody should tell the workers that you need to work. You need to bring food. We are hungry. Or you need to feed the larvae. Who does this job? In other species, it's the queen that does it, but here it does not. again you're stuck. So you go back to watching the wasp and you start thinking about it. And when we start thinking about it, it bothered me. The following thing bothered me. In other species, aggression is very important. So the queen is very aggressive, but other individuals are also aggressive. In fact, you can construct a hierarchy of aggression. You have an alpha, a beta, a gamma individual, most aggressive in rank 1, rank 2, rank 3. And if the queen dies, rank 2 becomes the next queen. But here, aggression is not used in this fashion. So I started wondering, what is the function of that low level of aggression? Why do they even have this low level of aggression where workers bite each other, chase each other, hold their antennae in the mouth? What is the function? What is the function of the low level of aggression? And I began to wonder whether it's a reasonable hypothesis that somehow this low level of aggression by the workers themselves is involved in regulating the activities of themselves. It is also known in that in advanced insect societies, there is such decentralized organization, not necessarily top-down. So I started looking for possible confidence in this hypothesis, possible ways of testing this. And then I found a very, so the question is, what is the function of the low level of dominance we have shown by workers? And I found that there is an interesting correlation, a very weak but statistically significant positive correlation between the fraction of dominance of all the aggressive acts that are going on, what fraction come to me? How many times have I beaten compared to all the beatings that happen? That fraction is positively correlated with how much food I bring. The fraction of aggression I receive is correlated with the fraction of my contribution to work. It's weak, but it is positive, it is statistically significant. So this allowed me to make the hypothesis that the low level of dominant subordinate behavior shown by the workers in a normal colony is used for the decentralized regulation of each other's work without any orders coming from the queen. Now, this is a nice hypothesis, but needs to be tested. If this hypothesis is true, we can make two predictions. If this dominance behavior is regulating work, then we should either be able to change the dominance behavior and show that there's an effect on work. I should be able to increase the dominance behavior and say more food should be brought, or decrease the dominance behavior and say less food should be brought. It is not possible to do this. As I told you before, lots of things come to your mind. Well, I don't know how to make the wasp more aggressive or less aggressive. But I can make the wasp less and more hungry. I can do the converse experiment. I can increase the nutrition level of the wasp and predict that dominance behavior should come down. Or I can make the wasp decrease the nutrition, make the wasp hungry and predict that the dominance behavior should. This I can do. And these experiments are done. These are very long and tedious experiments, but the fun of doing the experiment is so great that no student has ever complained that you have to sit for hours and hours and hours in front of the wasp and all. So in one experiment we showed that excess food actually reduces dominance behavior. Now, how was this experiment done? So my students learned to hand feed the wasps. We of course give them some food to eat on their own, but in addition, my students would take a piece of food. Now, food, by the way, is a caterpillar, is the larva of a moth. There is a very common pest of stored grain, a moth, which is called uh, Coursera cephalonica, and we grow these moths and we take these caterpillars, we feed to the larva. So my students learn to take a piece of the caterpillar in the tip of a forceps and quietly, quietly go and sit, hold it in front of a wasp. And the wasp initially would get scared, but after a while they'll come, examine, and take that food and run away. And you give them some more, and then you give them some more. So my student this way satiated the wasp. The wasp would then take that caterpillar, eat it, also give it to others, and feed it to the larvae. So my students did this till nobody was hungry and no wasp was interested in any more food. So this is how we excess fed. And then we measured how much dominance is happening. And it turned out that bringing food came down because we had already fed them. Feeding the larvae came down because they had already fed the larvae with the food we gave them. Most interestingly, dominance behavior came down. But this was not just non-specific dominance. It's not just that everybody was so full of food that they didn't do anything. Specifically, the dominance normally shown towards foragers who bring food, that came down. Food, those who bring food, they were not attacked. They were attacked much less than before. Gives you, but this is one half of the story. The other half is you have to starve the wasp and show that dominance goes up. This was actually easier to do because you didn't have to hand feed the wasp. We keep them in a cage. You just close the door of the cage and don't feed them. Starving the wasp is a bit easy, but the experiment, unlike here, where it was over 2 days, had to be done over 3 days. Because first day you have a control, you study everything, then you close the door, and that day you cannot study anything. Then third day you open the door and then you study, and we were able to show, in fact, that starvation increases dominance behavior. Again, you have day 1, day 3. So the dominance behavior initially, second day when there was no food, went up. Foraging, here foraging is the attempts by the wasp to bang against the doors of the cage and try to go out. They cannot, but they try to go out, and that increased enormously. And the food being, of course, there was no food brought on 2 days, but there was an increase in the food brought yesterday. But what happened was that the foragers who were bringing food yesterday were attacked constantly when there was no food in the colony. So we have reasonable evidence that worker dominance behavior is used for the decentralized self-organization of foraging by the workers themselves, not by the queen. Now this was very interesting because this began to suggest to us that the species we are studying is not a typical primitively eusocial species. So social species can be organized as primitively social and advanced social. The advanced social are the ants and the termites and the honeybees. The primitively social are the wasps and some other kinds of bees. And it looked like our wasp has gone one step beyond the primitively social species because this is a characteristic of highly used social. Honeybees do this, ants do this, but our kinds of wasps were never known to do this. So we already had one additional interesting result of this. Now, this is fine, but we already had worries about the meek and docile queen, because we also not only wanted to know how she manages to make the workers work, but how does she manage to make the workers not become future queens. So how does a meek and docile queen inhibit worker reproduction? How does she make sure that the workers don't start reproducing, don't develop their ovaries? This is also done by physical aggression in other species, and our species doesn't show aggression. Now here again, we have to think creatively how this might happen. But because in the previous result, we had the feeling that this wasp seems to do something that advanced insect societies already do, even though it is not yet so advanced. That gave us a clue. Maybe here also it is doing what advanced insect societies do. In advanced insect societies, obviously the honeybee queen cannot go and bite every worker. She has 60,000 workers. In ants, there may be 1,000,000 workers. In such advanced insect societies, the queens produce pheromones, and those pheromones inhibit reproduction in the workers. So you can think of it as a chemical castration. So these molecules are released from the body of the queen, they enter the body of the workers, and they actually prevent protein synthesis to make eggs, and ovarian development in the worker is suppressed in the presence of the chemical. You remove these chemicals, ovaries can develop. So we thought maybe that is what is happening. Maybe our queen has actually started producing a pheromone even though it is not supposed to do so. So we want to understand whether this is possible, and if possible we wanted to study this pheromone. But to do that you need to know at least something about this pheromone. The first thing we wondered, some pheromones are volatile. They just are in the air, and others are non-volatile. We call them contact pheromones. You have to go and touch them to receive them. So we wanted to know whether a pheromone that the queen might produce, is it volatile or non-volatile? And we designed a very interesting experiment to distinguish between these two possibilities. So what we did now was we brought a nest to the lab, put it in a cage, in a normal cage, and on day 1 we studied everything as before. On day 2, in the previous experiment, remember we removed the queen. Here we didn't do that. We actually removed all the adult wasps outside, and then we took a knife and we cut the nest in half. leaving approximately equal amount of larvae pupae on either side. So make them sort of equal halves. And once we did that, we inserted a wire mesh screen in the middle of the cage to separate these two nest fragments. The function of the wire mesh was to prevent the wasp from going across, to prevent non-volatiles from going across, but allow volatiles to go across. And once we had fixed the wire mesh cage, then we picked up each wasp, we tossed a coin, and we say heads you go to the left side, tails you go to the right side. Next was heads you go to the left, tails you go to the right. And since we don't believe in treating the queen any differently, we picked up the queen and we said same coin for you also, toss the coin, heads you go to the left, tails you go to the right. At the end of this we had the queen only on one side, one is queen right side and queen left side, and half wasp here, half wasp here, randomly distributed. Now on this day, we studied both wasps on both sides. And again, in our case, studied means calculate time activity budgets, calculate the frequency per behavior of all the wasps on both sides. And remember, in the previous experiment on day 3, we returned the queen which was removed on day 2. Here, the equivalent of that was we took the queen from one side and simply moved it to the other side, did not disturb the workers. So now yesterday's queen right side today became queen less, and yesterday's queen left side today became queen right. Why did we do all of these elaborate things? We had very clear-cut predictions. Our predictions were that if the queen pheromone is volatile, both nest fragments should behave like queen right colonies, because the wasps here will be able to smell the queen across the wire mesh just a few centimeters away. So nothing should happen. On the other hand, if the queen pheromone is non-volatile, very soon these wasps should begin to realize that they don't have a queen anymore, and they should behave like a queenless colony. What does that mean? That means one worker should become hyper-aggressive and try to become the potential queen. And if that happens, when we move the queen from left to right, this hyper-aggressive potential queen should go back to work on day 3, because now the queen has come back. And here one individual should become hyper-aggressive because now they have lost the queen. This experiment, as you can imagine, is rather difficult to do, because you have to put the mesh, you have to do all of these things. But this experimental design also turns out to be extremely powerful. So we call this the mesh experiment. And we have used this mesh experiment design actually to answer many questions. I will have one more question in this talk, I will bring back the mesh experiment. But because it is such a powerful thing, we have done this more than 100 times. Even when I made this slide, we had already done this experiment 24 times. And in all 24 times, prediction 2 was upheld, suggesting that the queen pheromone is a non-volatile substance. And I'll show you what the data looks like. In the absence of, this is on day 1, nobody is aggressive. Day 2, one individual becomes hyper-aggressive, and this is on the queen-less side. When I move the queen from that side to the other side, this individual drops her aggression, and a different individual on the new queen-less side becomes hyper-aggressive. Now we can go back and see what these individuals were doing beforehand. This individual was showing no aggression on day 2 because she was in the company of the queen. This individual stopped being aggression because now she is in the company of the queen. And now we can actually go back to day 1 and show neither the queen nor any of these individuals showed any real amount of aggression on day 1. In the experiment, since this one became aggressive first, we called her potential queen 1. And we called this one potential queen 2. And neither the queen nor the PQ1 nor the PQ2 showed any aggression one day. But exactly as our model predicted, this is what we saw on day 2. So the answer to our question is, Propylidia marginata queens appear to use non-volatile pheromones to inhibit worker reproduction, not physical aggression. But the fact that the pheromone is non-volatile kept bothering us. And the first question that occurred, right? Again, I want to emphasize that every time we have an answer, we don't have great confidence. We worry about it, and that forces us to answer the next question. But as we keep answering the question, slowly confidence builds up on the previous questions. So here we were worried. How is this? For example, we wondered, how do the workers actually perceive this non-volatile? in a normal call. Now, he said, let's begin with common sense once again. What is the common sense answer? The common sense answer is even though the pheromone is non-volatile, wasps move around, they interact, they touch each other, they bite each other. So every time they touch each other, there's a transfer of this chemical. So we said the potential queen gets the pheromone from the queen by physical interaction. That's how she gets it. And if she doesn't get it anymore, she realizes no queen and becomes hyperaggressive. Now, how to test this hypothesis? So what we did was we calculated by watching a number of colonies the rates of interaction, inter-individual interaction. So there are 20, 30 wasps in a colony. How often in an hour, how many times each wasp will interact, each wasp will touch each wasp. So we created a matrix of these interactions and we looked at how often the queen, the potential queen and the queen touch each other. Now I won't go into the details of this because that will take too long, but I just want to tell you that this question, which seemed to us a rather simple question, was one of the really tough questions. This question bothered us for 2 years and we almost had no answer till the end of 2 years. Because our common sense thing was that it is by physical touching and we showed that this is not true. And how did we show that this is not true? We found that the rates of interaction are too infrequent. The potential queens seems to realize the absence of the queen within half an hour of the queen's being removed, and they don't interact even once in half an hour. They barely interact once in a day. So it can't be. Then we said maybe the potential queen doesn't have to touch the queen. Maybe there are other wasps which are touching the queen more frequently. There are little other chamchas of the queen, and the potential queen has to touch these individuals and get it through a relay. So now we wanted to calculate what is the rate of flow of pheromone or information through relay from the queen to the potential queen. And this is not an easy task. Luckily, my neighbor's daughter, who was at that time doing BSMS in mathematics in IIT Kanpur, was actually my neighbor's daughter, and she had done some little projects when she was in school in my lab, and she had come home to visit her parents, so she just came to say hello, and I told her this problem. She said, I think I can solve this problem. So she was very good at MATLAB, and she used something called the Dijkstra's algorithm, which allows you to calculate the shortest path of information flow. So amongst all possible paths, so you basically write down every possible way in which information can go from the queen to the potential queen. One is direct, another is through one individual, through two individuals, through that. So you calculate all of that and you calculate the shortest path. And when she calculated the shortest path, it turned out even that didn't work. Even the shortest path took much longer than the potential queen was able to realize that the queen is gone. So we were dumbfounded and we were stuck. How is this possible? Back to watching the wasps. Now, one thing you must remember, that we all are human beings with a brain, and one property of our nervous system is called habituation. If you keep on watching the same thing, you don't see anything new. That is why you need students. That's why you need young students. You need students who have not watched wasps before. When students come to my lab, I forbid them from reading any of our papers or books. I don't want you to become a clone of me. Don't read anything, go and watch the wasp. For days on end, for weeks on end, marked wasp, we ask them, watch, 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 come up with your questions, come up with your speculations. Then we will channelize that into a research program. If you come and read all my papers, you become a subset of me, and you are then going to do what I might have done. What is the use of taking students if you are not getting new ideas from them which you cannot get? This time we were even luckier because there was an intern who didn't know anything. And he came and he was given a colony of wasps, marked wasp, watch. And he watched for a long time, great fun. Then he said, you know, the queen walks in some funny way. Well, we don't think so, but come show us. Then he actually demonstrated to us that when the queen is walking on the colony, she's actually dragging her abdomen. She seems to be rubbing her abdomen on the nest. And we immediately said, maybe the queen is actually applying the pheromone to the nest surface from where the workers get it. They don't have to touch the queen. And the paper is a substance which can easily absorb these chemicals. So it sounded like a very interesting idea. Before we went to the idea, these students intern project was decided immediately. Said we created a new behavior. I had only 100 and first behavior is rub abdomen. So his job was to study rub abdomen behavior. So he sat for days and days and days and measured the rate at which abdomens are rubbed on the nest by the queen, by each of the workers in the morning, in the evening, at night, on the front of the nest, back of the nest, side of the nest. So he had a huge amount of data on rub abdomen behavior. And the conclusion was only the queen rubs her abdomen. Workers do not do so, and she does it all over the nest. That gave us some idea. That seems reasonable to think of a hypothesis that she actually rubs her abdomen onto the nest, and that's how she gets it. So without going into the details, I'll just give you the conclusion in this case. The queen applies her pheromone to the nest by rubbing her abdomen, and that's where the workers get it from. But where is the pheromone? What is the source of the, sorry, what did I do? What is the source of this pheromone? How can you not wonder about it? How can you rest without answering this question? Now, I was neither a chemist nor an entomologist, common sense. What is supposed to be happening? The queen is supposed to be rubbing her abdomen. So I picked up a wasp and turned her upside down and said, what is there in the bottom of her abdomen? Turns out there's a hole. And if you dissect the wasp, there's a duct that goes from that hole to a gland called the Dufour's gland. The Dufour gland, by the way, is a very well-known gland in many social insects, has many different functions. But here it is coming outside like this. And we said our hypothesis is that the pheromone is made in the Dufour gland and released to the outside through this duct at the bottom of the abdomen, and that's how she rubs it. So we said, let's see if this is true. So this is what it looks like. So this is the Dufour's gland, and a duct carries it to the bottom of the abdomen. At this stage, we said to test this hypothesis. In fact, I do not like to do expensive experiments. I do not like to buy expensive equipment. But my students sort of teased me and said, now you have to buy a GCMS machine because you want to see what is in the Dufour gland. I said, no, not at all, not yet. Much more important than finding out what the composition is, you have to have a bioassay. Without a bioassay, there's no point in finding the chemical. You need a bioassay. So we designed a bioassay which killed two birds in one shot. The bioassay we designed was, we said, we will dissect the queen, remove her Dufour gland, make a crude extract of that, and we will take a colony where an individual has become hyper-aggressive. We know that if you return the queen, the aggression will come down. Now we will apply that pheromone to the nest rather than the queen and see if it comes down. If that comes down, then we have proof that the source of the pheromone is the Dufour gland. More importantly, we also have proof that it is the rubbing of the abdomen of the queen that is making the aggression come down. So it is reinforcing our hypothesis that the queen uses the non-volatile pheromone, plus telling us that the Dufour gland is at least one of the sources of pure pheromone. And of course, the control experiment is you also workers also have Dufour gland. You extract those glands and you should show that aggression does not come down if I apply workers Dufour gland, and aggression does not come down if I use a ringer solution which I used to make the extract in the first place. That's exactly what we found. First session, no aggression because the queen is there. Second session, queen is not there, aggression has gone up. And third session, when I or my student applies the pheromone, there's a significant decrease in the aggression. No such decrease with worker Dufour gland, no such decrease with applying the ringer's pheromone. So we had confidence in two of our answers, that the Dufour gland is at least one of the sources of the queen pheromone, and that the queen pheromone is responsible for the presence or absence of the hyperaggression. So then we had to look at what it is. Then of course we went to a neighbor, a kind neighbor said, you have a GCMS machine, tell us what is in this. And for a short time, some of my students and I had hopes that we will find the pheromone. We'll find a molecule, put it in a wire and show it to everybody. This is the queen pheromone, and then we will synthesize it chemically and we'll do all kinds of things. But of course, luckily, that didn't happen. And I'll tell you later on why luckily it didn't happen. It actually turns out there are at least 30 different compounds of different amounts. The interesting thing is that these are the compounds, and it is the area under the curve that is different between queen. There is no compound that is unique to the queen or to the workers. Nothing is uniquely present or absent in the queen and workers, but the cocktail is different. The area under the curve is different. So by measuring the area under the curve, we can distinguish between queen and worker. What do we mean by we? That is, our computer can distinguish. If you supply a computer the information about the area under the curve, each of these, it will correctly, with 100% accuracy, classify queens into queens and workers into workers. So there is enough information in the area under each curve for a computer to be able to discriminate between a queen pheromone and a worker pheromone. There is enough information in the pheromone for the wasp to know who is the queen and who is not the queen, but we don't know. We cannot tell. So the next thing I'm going to tell you about how we cannot make this distinction. So clearly, we suspect that the source of the queen pheromone is in her Duffer's gland. Notice the word suspect. Caution is very important. How can I be sure that Duffer's gland is the only source? That's one of the sources. It could be in other places too. So, but next question is, it's also important where the questions come from. We often think we write scientific papers in a very, very artificial way. We So in this case, how do we proceed? What's the next thing we should do? So in this, what happened? So our questions, as you already seen, come from various places, often from dissatisfaction with the answer. But one question came from the audience. So every time I discuss these results with audiences like you, people were struck by many things, but in particular struck by the fact that you remove the queen, one individual becomes the next queen immediately. Who is this individual? As human beings, we are of course extremely interested in transfer of power. Who is the next director? Who is the next prime minister? Who is the next president? We always want to know. So everybody wanted to know, how can you predict who will become the potential queen before removing the queen? And initially I say it doesn't look like it. There seems to be nothing particular. I don't think we can predict. But this question became persistent. And while I said I must do something about this, I can't just casually say not possible. So by then I was a full professor, and for a full professor, do something means the next PhD student. So when the next PhD student came to my lab, I said, your job is to predict the identity of the potential queen before removing the queen. You got a PhD. She is thrilled. She said, oh, wonderful. I am honored to have this responsibility. She worked for 5 years, did lots of experiments, published many papers, but she threw up her hand and said, I cannot predict the identity of the potential queen. We gave her a PhD, all right, but the answer is we cannot predict it. And for example, she showed, can we predict? The answer is no, because the potential queen is mixed up amongst all the workers. These are queens, these are workers. So the potential is not unique in any way. We cannot predict who she is. I actually sort of was happy about this, because I had begun to love these wasps so much that I felt they know there are some secrets which they are not willing to yield to me. They know something which we do not know. The next student, dynamics between students, between success students, is extremely interesting. When we write about our research, even when we write our autobiographies or biographies, we don't bring enough of this. It's very, very important. Students learn from each other, they are jealous of each other, they are envious of each other. All of this is very, very important for science. So the next student came to the lab and said, yeah, this is all fine. My seniors have done all of this. Okay, we don't know. But I suspect, again, she had watched the wasp for a long time by then, she said, we don't know who the next queen is. But I strongly suspect that the wasps themselves know who the next queen is. So the next question is, do the wasps know who their next queen is? This Seemed like a very difficult question, and we were summarily discouraged by all our friends and colleagues. They said, you cannot do this because this is not a scientific question. How would you know the mind of a wasp? How would you know whether a wasp knows something? So we were actually discouraged. But this student, the more you discourage them, the more she said, no, I'm going to find out. So we actually spent 2 years discussing on the blackboard, thinking of ways in which we can answer this question. And after some time, we came up with an experimental design which we thought will answer this question. And we did the experiment and we got the answer that they know. But very quickly, we realized that there's a flaw in that experiment. It's very important for you to kill your own theories, to kill your own result before the referee does it. And we realized that there are alternate explanations for this. So we went back to the blackboard, but eventually we came up with an experimental design which I think will allow us to answer, and I believe that we have answered, and I'll illustrate that experiment in the next few minutes. Here is where I bring back the mesh experiment. But the way in which we did this experiment was totally different from all previous experiments. In previous we had a question or we had a hypothesis. Here neither question nor hypothesis, but we had an assertion. We said so first we said let us, okay, so we cannot identify because not unique. Now we said let us first frame the question in a more technical way so that people are more sympathetic to what we are doing. So instead of saying do the wasps know, simply we said is it possible that there is a hair designate even though we cannot identify her, and do the other wasps somehow know who this hair designate is? So the philosophy of this piece of research was as follows. We are going to assume that there is a hair designate. We are going to assume that all the wasps know who they designate. We don't know, but they know. Now we are going to do an experiment and make a prediction of how the wasp should behave if our two assumptions are correct, and how they should behave if our assumptions are wrong. Okay? So what is the experiment? The experiment is bring a colony to the lab, cut it in half, have two halves, one side queen, less side. Now imagine what would happen. Most of the experiment should be first done in the head, then only in real life. So think about it. So what do you think will happen? So this is the experiment. This is one half, this is the other half. This is the queen. Now there is only one hair designate, and we already assume there is a hair designate. Now by chance alone, this hair designate could be on the opposite side of the queen. Entirely possible. Now, if this happens, what would you expect? Surely this individual will become a potential queen because we already know this. But this individual is supposed to be the potential queen for the entire colony, not just for her side. To test whether she is indeed acceptable to all the wasps in the full colony, what we do, we move the potential queen to the left and the queen to the, we exchange position. And we expect this potential queen to be respected even on this side. Move her back, there should be no problem, because she is supposed to be the hair designate for the whole colony. However, such a nice turn of events can only happen in about half the experiments, because by chance alone, in the other half of the experiment, the unfortunate hair designate is sitting next to the queen and she cannot do anything. Now, what would you expect would happen? Here there is no hair designate, no queen, but perhaps the best individual here will become the next potential queen. So if she does, let us call her potential queen number one in this experiment. But exactly the opposite of the previous argument, this individual is the best only for this half. She is not for the whole colony. The true hair designate is here. So what do? We bring this false potential queen face to face with the true hair designate. And we expect that all hell will break loose. Unfortunately, all hell did not break loose because this individual voluntarily gave up. And the hair designate became hyper aggressive. But that's not the end of the story. This individual, the acid test for this individual is usually acceptable to the opposite side. And this is what is our prediction. So what is our prediction? We have the following predictions. First is the first potential queen should be unacceptable to the opposite side in about half the experiment. And a second one should emerge in those experiments. However, there should never be a third potential queen. The second potential queen should always be absorbed both sides. There should never be 3 contenders to the throne if you cut a nest in half. These are our predictions. Now, as you can imagine, these experiments are even harder to do. It's not the student who gives up, but often the wasps give up because it's too much disturbance. But this student was so invested in this that she spent another 2 years, finally managed to do this experiment, seeking the cooperation of the wasp with all her skills 8 times. In the first 3 experiments, she found that the first potential queen was successful on both sides. She was not challenged at all. This corresponds to a situation where the hair designate by chance happened to be on the opposite side of the queen. But in the remaining 5 experiments, the first potential queen lost, a second one came, but she was successful on both sides, corresponding situation where the hair designate in the beginning was on the queen side. And this is what the data would look like. This is what the data would look like. Day 1, of course, nobody is aggressive. Day 2, there is one individual who is aggressive, and day 2, there is a different individual who is aggressive. So here, this is the 3 experiments in which the same individual, and then she changed. So in summary, we can say that a potential queen 2 emerged in about half the experiments, and no potential queen 3 ever emerged. So there is a hair designate. But we want to go beyond that. We want to say there is a hair designate and all the wasps know who this hair designate. It sort of appears to be so, but the confidence, our confidence in. So yes, so there is a hair designate even though we cannot identify her, and the wasps seem to know who she is. Our confidence that all the wasps know this came not from the data, not from the statistics, but by a simple observation. What did we find? We found that, remarkably found, that the potential queen number 2 was never challenged. She did not receive a single act of aggression by the potential queen 1 or by any of the workers, either when she emerged on the side or she went to that side. And that is why we have confidence that the hair designate was obviously known and acceptable to all the wasps, including the potential queen 1. She knew that's not my turn yet. That is why we have confidence. This was a very interesting period for me because I used to be asked, do you know who the potential queen is? And I would say, I don't know. Now I said, I don't know, but I know that the wasps know. We know that the wasps know. Indeed, we were allowed to publish a paper with this title. We know that the wasps know because there are cryptic successors to the queen in Rhopaladium argenata. So there is a hair designate, even though we cannot identify her, and the wasps seem to know. Sure enough, the next student who joined the lab was not impressed. He said, yeah, that's true, but how does she know, that is his senior, that there's only one hair designate? Maybe there are many hair designate. Maybe they're all standing in a queue. Let's do the experiment. So the experiment here is to take a queen, observe it, identify the queen, remove the queen. Within half an hour, you identify the potential queen, remove her and see what happens. And he did this experiment, and he showed that if you remove the potential queen, there'll be one more. You remove her, there'll be one more. In fact, the data looks like this. Is there just one hair designate, or is there a queue of hair designate patiently awaiting their turn? Yes. Normal colony, nobody is aggressive. Remove the queen, it turns out that one only and only one individual will become hyper aggressive. Nobody else. Remove her, of the remaining individuals, one and only one individual become hyperaggressive. Remove her, one and only one individual. Remove her, one and only. And he did this for 5 potential queens, and he showed that I have proved the proof of principle that in fact there is only one. And I want to blow up this here and show you that this one and only one individual shows aggression but doesn't receive any aggression. They are not challenged. I have enlarged this here to show you that these blue bars, which are supposed to be data on aggression received by the potential queens, which are essentially 0. But this is what they show. So indeed, there is a long reproductive queue of at least 5 potential queens, but perhaps the queue is even longer. Perhaps everybody is standing in the queue. Now you might ask, and you should ask, if there is so much peace, so much cooperation, so much order, where is the war? Where is the conflict? Indeed, I must confess that we were from time to time very disappointed that there is no conflict. There is no severe aggression. We were itching to see a fight. We were hoping that finally we move PQ to this side, PQ1 and PQ2 will fight. But even they didn't fight. Why are these wars so civilized? But we want to see a fight. So now we said we are deliberately going to do experiments where we want to see aggression. We want to see fights because theory tells us that there must be conflict. These wasps are not clones of each other. There must be conflict. How to find that conflict? So we did some experiments. We asked, how does the queen maintain her status and why does she lose it sometimes? Because we know in nature from time to time the queen is replaced by another worker, maybe because she's old or whatever. But until then she maintains her status. In the presence of the queen, nobody ever lays eggs. But remove the queen, immediately there is another individual. So how does she maintain her status until some point in time, and why does she lose it sometime? How to test this? So it seemed to us that the best way to test this is to prevent the queen from applying her pheromone to the nest. If she cannot apply pheromone, workers will think there is no pheromone, so there is no queen, and this individual is some imposter, and they will chase her out. And a new individual becomes putting queen. Just like the wasp on the other side of the mesh when there was no pheromone, right? So how to prevent the queen from applying her pheromone? So people said you block that opening in the abdomen. In fact, some people said you apply nail polish to that. I don't like to mess with the wasp so much because I think that will be too disturbing. So I thought for a long time I came up with an idea. So what we did was we went to nature, studied a colony or several colonies in great detail, identified everything. Then we collected all the wasps, not the nests, only the wasps, and put them in a big cage. Now we said, come on, queen, where will you rub your abdomen? There is no nest. And we expected that she will be overthrown. So we repeated this experiment 6 times, and we found to our great surprise that the queen was not overthrown. How come? When we looked at our observation in great detail, it turned out that the queen was frantically rubbing her abdomen all over the cage, although there was no nest. And that is why she was not overthrown. But we were not satisfied with this explanation. We said the same amount of pheromone, if you apply on a 2-3 square centimeter nest versus the whole cage, it must be very dilute. And the workers must sense this. And indeed they sensed it because they attacked the queen. Never in a normal colony we have seen workers attack queen. They attack the queen. But then why was the queen not overthrown? Even greater surprise, the queen fought back. The meek and docile, non-aggressive, non-interactive said, yes, I can fight. So when required, she can fight. Indeed, that's what happened. The queen continued to rub her abdomen on the walls of the nest, but the workers attacked her, which they never do in normal colonies, and the queen retaliated. She almost never does this in normal colonies. There is potential for conflict, but it is usually prevented from coming outside. And that is why I think these wasps are so successful. There is always potential for conflict, but they are able to manage their conflict very well. And that is what I find most impressive. But we wanted to see a little more aggression, and we said if to do that, what we need to do is to look at the interaction of wasps with outsiders. So how do the wasps react with wasps from outsiders? And so here we did an interesting experiment. We had one cage, we had 2 cages, one nest here, acclimatized, studied, everything fine. Then one day we took all the wasps from one nest, no nest, only the wasps, and we introduced them into this cage. So now in this experimental cage, we had a resident nest, resident wasp, and alien wasp without any nest flying around. And we wanted to know how the residents will treat the aliens. And we found to our great surprise that the residents treated the aliens in a very nuanced and sophisticated manner. It was not a blanket thing. Let me summarize what they do. When the members of one colony were introduced into the cage of another colony, young alien wasps were admitted freely into the colony. No problem, just come and join us, you're welcome. Older workers are allowed to live in the nest, but don't come anywhere near my nest. You want to be in that corner, fine, but don't come near my nest. And the alien queen was located and physically torn to pieces. So a very nuanced treatment of the residents depending on this. This was very interesting, very satisfying, but it gave us a new idea which we had never thought of. If young aliens are accepted, we can now make genetically mixed colonies. So we went to Mysore, collected wasps from there, brought them to the lab, and when the young ones we put them in Bangalore colonies. And now we had genetically mixed colonies. We could study these genetic mixed colonies. We answered many questions with this, but one question I want to talk about today is what is the fate of these young accepted aliens? To our great surprise, we found that young alien workers who are accepted into foreign colonies become completely integrated into their foster colonies and become normal workers and may even become future queens. Indeed, we found that the probability that a foreign worker will become a queen is directly proportional to the number of foreign workers. No distinctions once they get integrated into the colony. That is War and Peace for you. The wasps have a great propensity to make war with outsiders and an equally great propensity to maintain peace with insiders. But as an evolutionary biologist, I will tell you that from an evolutionary point of view, war with outsiders is easier to explain than peace with insiders. But war with outsiders is not of much use unless one can combine it with peace inside. And it is this dual strategy, this ability to tread a fine balance between conflict and cooperation that I think accounts for the success of insect societies in general, and that is why we call them superorganisms. So we had to extract war out of this. I want to close by just pointing out that if you want to read more about this, there is a book called Survival Strategy, which I have written many years ago, and there is a book called on Rhopalidia, which I have written more details, but also I have written a book called Experiments in Animal Behavior, cutting edge research at trifling cost. Is freely available. As you have noticed, most of my experiments did not require any sophisticated equipment, any great laboratories, any great amount of money. Mostly it was thinking and doing things in a simple way. Many experiments of that kind have been done, and anybody who finds that they don't have enough money should not say I can't do any good research anymore. There are areas in which you can do research with very little money. I have discussed this in this book. I also write frequently for the general public. You can look at this. I want to close by thanking, by acknowledgements. Yes, it is true that I have received funding from many government agencies, but I didn't need much money. As you can saw from my experience, I need some wood and some wire mesh, and that's all I need. In the old days, I used to buy my students laptops. Now they come with fancy laptops of their own. I don't need to do that. So most of my money I use to pay salaries for my students. I much prefer to work with students than with machines, and these are my students. I want to thank with them. All of them are passionate watchers, and they all have contributed to research, and I am the spokesman telling you all the things they have done. Thank you very much.